All right. Hello, everyone. Good morning, good afternoon, and welcome. Today we've got a pretty action-packed calendar, I think. NCBI has graciously agreed to give us an overview of where they currently sit with STAT and some of their SRA submission processes, and some eagerly awaited updates on not only hopefully the SRA side, but also potentially some of the local host removal tools that are available. And Jeremy and Emma and Noah and others have agreed to give us a brief overview of their recent paper on 677 variants. So we might get into a little bit of ornithology while we're there. Before we go on, though, I wanted to, so there's the agenda for today, give a quick update on in terms of numbers. U.S. submissions right now are at 110,269 in GISAID. That is an increase of about 10,000 over the last week. We've had some minor hiccups in getting some of the commercial sequence data into GISAID and GenBank, but I think a lot of that has been resolved over the past couple days, I would think, through diligent efforts on all fronts. But I think that should resolve itself fairly quickly, and we should see an uptick in both pretty dramatically as some of these large sequencing efforts start to come online. In terms of GenBank submissions, that translates to about just over 41,000, 41,420, which is just shy of 3,000 since the last update last week. Hopefully, we will start tracking SRA submissions pretty soon because I think that's going to be an important addition as well. But obviously things are starting to increase pretty dramatically, and I would imagine that things will continue to accelerate as submissions continue to increase. Just in the interest of time, I'm going to kick it over to Chris O'Sullivan and Yuri and whoever else from NCBI is going to drive today. I'm really looking forward to this update and can't wait to see what they've got in store. Chris, over to you.
Hi, thanks. I'm broadcasting live from my fabulous basement office. So my lighting is terrible. Apologies if I look like a psychopath. Anyway, I'm here. I've got my one of my developers who's going to do the bulk of the technical talk for us. I'm going to introduce it by just talking about why on Earth NCBI decided to develop their own human read scrubber, which was basically it led to the development of our taxonomy analysis tool. And we have just submitted a paper on that describing the full technique and methodology. I believe there may be a bioRxiv link which will be available in the talk coming up shortly from the developer, who I'll introduce shortly. I just wanted to give the background that back in the beginning of the Human Microbiome Project, there was some concern that some of the microbiome data would contain some reads from the host. A lot of people thought, oh, well, we're just doing PCR amplicons at the beginning of the project. Why would there be any human reads in there? Well, there are a lot of them. So we had, at NCBI, we've got a fantastic developer. Her name is Risha Agrawala, and she had been working on building an alignment tool. And she quickly adopted that to create a branch that would find human reads within an SRA data set, flag them, and then SRA would blank them out. And this worked fantastically for the Human Microbiome Project, but that worked in the specific context of HMP submissions, which, because of the nature of the project, they came in as dbGaP projects, meaning they came in with a lot of host phenotype data. So they had to go through the dbGaP submission process. Now, when NCBI first stood up the dbGaP portion of SRA, we actually created a physically isolated file system just to make sure that we're not leaking, any personally identifiable sequence information, and that it was inaccessible to anybody who should not have access to it. And so these HMP submissions would come in, and we would run a human, an alignment against the human reference genome, and then create the filtered copy of a file to copy into the public side of SRA for distribution of just microbiome data. A few years later, we started working with the FDA and CDC on foodborne pathogen surveillance. And there were some requests from some potential submitters about possibly applying a human filter to their submissions. Now, since these were not dbGaP submissions, they would be coming directly to public. And on the public side, just the technicalities of the way NCBI processes data. SRA is one client of very many using a shared compute resource. So we realized that these jobs were not, these human scrubber jobs were not getting queued efficiently or quickly. Basically, a human reference index is a 15 to 20 gigabyte file. And when you're requesting resources on a shared compute resource, you have to request memory, and then you have to wait for a host to be available. And then you have to move the file. And it's not very fast to move a 20 gigabyte file. So right about that time, serendipitously, we had a new developer join the team. He just left Microsoft for reasons that I'll leave alone. He had no background in bioinformatics, no background, not really much background in biology. And I thought, well, maybe this is perfect. We'll just take a completely fresh approach. I'll just present him the issue, and we'll figure out a way to make a smaller reference index. That's my big, dumb biologist approach to things. Like, well, there's got to be a way to make a smaller index. Like, we don't actually need reference placements. We don't need to know where in the genome these things align. We just need to know whether or not it was human. And so after a week or so of discussing the issue and talking to this new developer, Oleg, I taught him a lot about biology, a lot about microbiomes, a lot about DNA structure and sequence analysis. I mean, just the basics, as much as someone could learn in a week. And he said, I think I can go figure something out. My suggestion was, well, let's just start deduplicating the human reference. Let's throw out the low complexity sequence. Maybe we'll make a smaller index that way. And well, after a couple of weeks, he came back and he said to me, we can do this. We can reduce the size of the index by 50-fold or maybe more. And then he said, and I think, basically I said, all we need to do is find reads that are definitely human and definitely not something else. We just want to eliminate the false negatives. And he said, yeah, we could probably do this for all organisms. But anyway, this was the start of what became our taxonomy analysis tool. And I would like to turn it over to another developer on our team, because unfortunately Oleg had to leave NCBI. He took a job with SpaceX, which is a typical fate for many NCBI developers. He's actually rejoined us recently, but I'd like to introduce you to Ken Katz, who has dug into the tool, sorted out the code, and actually written it up as a publication. And he's going to walk you through the technical aspects of how this thing actually works.
Thank you, Chris. I really appreciate that. And first, let me apologize. You notice I'm not sharing a video, which was my bad in terms of setting up my Zoom. But then I realized, because many in this audience are from the CDC, it's really bad. Many of you may know a Kenneth Katz, Kenneth A. Katz, who I know, but who I am not. Ken used to work at CDC. He's a dermatologist, and his father, Steve Katz, his late father, Steve Katz, was a colleague of mine because I'm trained as an immunologist. And I didn't want anyone out there thinking that Ken had returned yet again to the DC area. He's still out in California.
Apologies, and I meant to introduce you as my favorite type of developer, the biologist who decided to take up programming instead.
Thanks, Chris. So what I'm going to do, first of all, is just introduce you to some of the key concepts of our NCBI stat tool, the sequence taxonomy. Come on, you can say it. Analysis tool. And the reason we call it stat is because I like to be able to breathe when I'm talking about it, and that's a mouthful. Now, stat really refers to a collection of tools, and these collection of tools are for building Kmer databases, for querying those databases, for reporting the results of those queries. And what they have in common, what they share in common are two key concepts. And these two key concepts are what allow both stat and the human scrubber, which is based on stat, to be accurate and fast and scalable. So the first key concept is called Minihesh. Minihesh was developed by Andre Broder back in the early days of the web. And it's really a deceivingly simple concept, but it is the key concept that allows stat to be scalable and fast. And the Minihesh principle simply states that a random selection of the lowest valued constituent blocks in a pool after hashing represents a signature of the parent object. Don't worry about that big sentence because I'll break it down a bit. What Broder was concerned about back in the early days of the web, if you can remember through those misty vision in your eyes, the web viewer browser Alta Vista, they were concerned about how do I tell whether two web pages are the same or are different. Now, I was not imaginative, and in the 80s when I was staring at my ARPANET screen, if somebody had asked me that question, I would have said, I'll look at the two pages and I'll tell you if they're identical. And what people like Broder realized was that that's not really a scalable method and it's not going to work. And they presciently saw that there was going to be an explosion in web pages. So you really needed something better. So the idea here, again, first we're going to take a random selection of the lowest value constituent blocks. So if a web page is a bunch of words, say 10,000 words, what we might do is have a sliding window of 3 or 4 words, and we're going to slide that window through the document. And now we need to assign a value to that window of words because we want to find the lowest value of those words. And so maybe you might assign the numerical value to a letter of its position in the alphabet. A equals 1, B equals 2. That's about as ingenious as I can get. So you can imagine now sliding that window, generating values for each of the words and the letters that make up those words in that window. And now you simply select the lowest valued constituent blocks. And because this is a sampling method, you may be comparing two web pages that are 100,000 words each. But in fact, you could just grab a handful of these lowest valued constituent blocks, maybe 10 each, and by this principle, if those two handfuls were similar, it would tell you those two parent objects are similar. So now you can immediately see that this is scalable, and where things are scalable, they become very, very fast. One thing I should point out is Minihesh is widely accepted. It's used in not only in many areas of bioinformatics, but more famously, because it was based on words, it's used for ferreting out plagiarism. Now, in SRA, our parent object is, of course, a nucleotide sequence. And so when we implement Minihesh, what we call the constituent blocks are Kmers. And a Kmer is just a polymer of length K, and our blocks are nucleotide polymers of length K. And what we're going to say is from a set of sequences, we're going to read a window. Our window is going to be 32 base pairs. So these are 32 base pair Kmers. Those are our constituent blocks. And we're going to read them across a window. And then we're going to transform them into a 64-bit hash. And just as I described for the original Minihesh, we're going to select the minimum valued Kmers, and they are going to be representative of that parent set of sequences. And I would point out, crucially, that for every Kmer that we ultimately select, we store the tax ID for the sequence it was derived from. Now, let's just focus a bit on how we really do a Kmer Minihesh selection. In panel A, I just show a random sequence. And above that sequence, I've shown a bracket for a 64 base pair segment. Below that sequence are another series of brackets. Those brackets represent 32 base pairs. That's our sliding window. And what I'm showing below that sequence is we are going to slide that 32 base pair window, incrementing one base at a time across that 64 base pair segment. And so what we're going to do is generate 64 32 base pair sequence Kmers. And then from those 64, looking at both strands, we are going to apply our hash and then select the lowest valued Kmer. Now in panel B, we can look at what we really mean by creating a hash. So first I've shown a 32 base pair sequence, just randomly from that original 64 base pair segment. And the first step that we do is we turn that 32 base pair sequence by encoding each base in 2 bits into a 64-bit binary array. This is a really simple code, and it's very standard in bioinformatics. So instead of each nucleotide taking up 8 bits or 1 byte, each nucleotide will now take up 2 bits, and that's a 4-to-1 compression. And what I'm showing below that 32 base pair sequence is literally the 64-bit binary array that represents the sequence above it. And now the real trick that Oleg imparted to us was all you got to do is read that 64-bit binary array as a decimal. So now if we do this very operation for all 64 32 base pair segments that we've generated in this window, we simply read the decimal value of that 64-bit encoded sequence and choose the single lowest value. That's our min-hashed Kmer that's going to represent this 64 base pair segment. So that's how we implement the min-hash selection technique. And again, that's really a key concept because it obviously allows us to be scalable. When we're selecting 1 out of every 64 possible Kmers, that means the size of our Kmer database is much, much smaller. The amount of memory that you use, much, much smaller, etc. However, as I also said, there were really 2 key concepts to stat, and Kmer hashes by themselves do not make a database. And the real key observation here is that a result of evolution, as we know, is that for any given species, most of the sequences in that species are not specific to the species, but they rather represent that lineage from which that species arose. we all know that humans mostly have primate and chimpanzee, even some gorillas, certainly Neanderthal, we now know, sequence. And so our task here, the second key in making an accurate database, is that for any given tax ID, we need to identify those Kmers that are diagnostic for that tax ID, for that species. And in that process, we can then move all the other Kmers up the lineage to their lineage-specific appropriate node. Let me just give you a small cartoon of how we do that. In panel A, you can see depicted a genus and two species. And what you can see in panel A is that species A and species B have some Kmers unique to themselves, but they also have two Kmers that they each contain, and those are indicated in bold. And the idea here is we move to panel B, where we have taken those Kmers that are not diagnostic for either species A or species B. We assume they are diagnostic for some other node on that lineage, and we just move them up to the genus. And now we have only specific diagnostic Kmers for species A and species B. And the key here is we iterate this process starting at the very leaves of a taxonomic tree, a bunch of species out to your right or your left, depending on whether you're left-handed or right-handed. And we just start at the species and do this operation up to the next level, which might be a genus or subgenus, and then iterate again and iterate again. And we do this merging all the way up to the root of the kingdoms. That's the step that makes a stat database not just scalable and fast, but accurate. Now, let's get to the human scrubber. And Oleg, as Chris indicated, made a really special human scrubber database for this purpose. What he started with was a green bucket on the left, in which he generated representative Minihesh Kmers from all human RefSeq sequences. Now, what's key in that green bucket is no merging has happened. So in that bucket are all the human-specific diagnostic Kmers, as well as human Kmers that represent lineage. What we're going to do is subtract from that green bucket the blue bucket, which is all of non-eukaryotic Kmers. And just to make this easy, we use the non-eukaryotic Kmer merge tree that we generate during our normal stat building process. So if we subtract the blue bucket from the green bucket, we're left with the yellow bucket. That's our very special human scrubber Kmer DB. And this DB has the following two characteristics. It is very aggressive at identifying a potential human read. It's aggressive because not only does it have those diagnostic human Kmers in it, it also has Kmers that humans have because they really represent primate lineage or mammal lineage or any other lineage up the eukaryotic tree. But the other key is it is very conservative at maintaining any potential viral or bacterial clinical pathogen sequence. So that's what we want. Aggressively remove a potential human read. Do not touch the potential clinical pathogen sample read. This is the best slide, and I really appreciate all of Oleg's work because this is all you have to do. It's this simple to scrub. You have an input sequence, which might be input FASTA-Q. That just means this is the FASTA-Q sequence. The FAST-Q, I always say FASTA-Q, excuse me. The FAST-Q sequence, which was the output of your sequencing run. and you hand it to the scrubber. The scrubber is just going to chop it into 32 base pair Kmers. It's going to align it in some sense with the Kmers in this very special Kmer database. And what it outputs is a new FASTQ file, and that is your original FASTQ file minus any of the spots or reads that had potential human PII. It's really that simple. How good is the scrubber? This is a very key observation by Oleg. So using this example, excuse me, of a run, this record is actually one of the first clinical samples from Wuhan, and it's bronchoalveolar lavage. Just a little washing of bronchoalveolar tissue. And lavage expects to find mostly lymphocytes in an infected patient with some unknown pathogen, epithelia cells, and of course, hopefully a viral pathogen. So this run had over 5 million spots, and this 5 million paired reads, essentially 1.5 gigabase pairs of sequence. And the read length is variable, as it typically is, reads going from less than 80 base pairs to about 150 base pairs. Actually, in this case, I think even some up to 170 or 80. And we knew from the stat analysis that of those greater than 5 million spots, 91.54% of those spots were eukaryotic. So from the point of view of our human scrubber, that means there should be 4,796,442 human spots in that run set. So when we ran the scrubber, we removed 99.97% of those human spots. And it's in yellow on this slide because that is really impressive. 99.97% of human spots were removed. Of the 0.03% that remain, they tended to be, in fact, in the cases I looked at, they were always short reads and typically of low complexity. These are not reads that are going to contain any PII information. So as you can see, the slide says a little demo, and I believe there's enough time for this simple demo. I'm just going to show you that I'm starting with a local file. That's a FASTQ file. And I'm now going to execute this command, which is just running a Docker file. This Docker image is public, by the way. It is sort of a beta version because I have not had time to put a test in it, but you can try it. And we do have on the slides, we've put its location. And there I am running the Docker image. I'll just point out that the first part of that command is really standard Docker verbiage. You're just passing the Docker image your present working directory. And you're allowing the Docker image to read it locally and write to it locally. And the reason that's important is because your FASTQ file is not in that Docker image. And the file that it writes, it doesn't write to the Docker image, it writes to your local directory. So it's finished, and it's just cleaning up now. And we see that it took about 38 seconds to run through. I think in this case, there's about 400,000 reads. And now what I'm going to show you is, in addition to the original file, which is the one that we used as input to the scrubber, there's a new file. And that new file has the same name as the input file with the added extension clean. And if you look at the size of the files, you can clearly see that the original file is a bit bigger than the clean file. That's because what Scrubber has done is it's removed just about 139 spots, if I'm not mistaken. And what we can do is look at some of those differences. And all I'm going to do is take this spot, or this read. This read was removed from the original FASTQ file because it was flagged as human. So let's take that. We paste it in this window. Okay. I think that's the right sequence. Let me make sure. I missed some. Let me get the whole sequence. I hate to miss a few bases. All right. So now I am going to actually go back to my save. I'm going to view. There it is. And we're just going to blast it. And so this is blasting a 194 base pair read. And we can see without doubt that that read was correctly flagged as human. Some really nice identity there. So that's just an example of how we use the scrubber. And this is, of course, just an example of how good it really works. I can tell you that I had randomly selected for this demo 2 reads. They were just the first 2 runs, just the first 2 runs I could find that had SARS-2. And in both cases, submitted from state labs, the scrubber found very convincingly human reads and removed them. And these were important reads to remove because in at least one case, even I would have had no trouble probably finding PII in it. So with that, let me go back to the presentation here. I will say in the presentation, I have screenshots of the demo I gave, so you can see the commands that I executed and the example of the output. And now I'm going to hand it back to Chris, who can kind of wrap it up. So go ahead, Chris.
Oh, am I back on? All right.
Yes, you are.
Right. So thanks, Ken. I hope that technical explanation was helpful for a broad audience. Basically, we just want to point out that we have built a fast and extraordinarily efficient tool that can be used scalably on shared compute resources or in the cloud or anywhere you really want. It is extraordinarily specific for identifying reads that are going to definitely belong to a given species. We've also made sure to not exclude reads that should not be excluded. We're looking at, essentially microbiome samples, viruses, or bacteria. And so our human scrubber database has subtracted out any Kmers that might be found in, consistent with bacterial genomes or viral genomes. So we're not going to remove things that should not have been removed. But the statistical analysis shows that it is extraordinarily successful at identifying any read that might actually be of human origin, particularly ones that would possibly contain identifiable SNPs or identifiable sequence. The method works. It's successful. You can compare it. Yes, if you use our tool and scrub a read, a given SRA run, and then say, oh, I'm going to align this to human, you'll find that some things align. But if you decide to align it to mouse, those same reads would align. Basically, we're removing everything that could be human, and we have not found any evidence that we're missing something of significance, nor have we had any complaints from anyone who has used it. I encourage you all to check it out. We've got links. It's effective and it's fast and it works.
Hi, Chris. I'm just going to take that and just say a few things quickly. First is, while we do have the manuscript that has been submitted for publication, and I did submit it into bioRxiv, but it's still in the screening process, and I haven't had my email on, so I don't know the status of either of those. I had that email off because I didn't want to be disrupted when I gave the talk, so I apologize for that. The second thing I would like to point out is I was terrible. I totally disregarded the instruction I was given, and I ignored the chat. I'm happy to answer any questions at all. Some of them are very, very good. I don't know if you, I'll let Duncan decide how to do this, I suppose. I don't know if you want me to look through for the good ones. Duncan, you pick the good ones. I will also say anybody is welcome to send emails or contact me by any means. I'm happy to talk about it. I think that's about it, except probably the most important slide, which is that one. There are many more people than this who have worked on the scrubber, of course. The most important in some ways is the last, Oleg Shutov, who did a lot of the great work. I want to thank everyone who helped me in all my endeavors, writing the paper, understanding the code, pursuing these wonderful tools that we can share with our colleagues. So I think with that, I might hand it back to someone else.
All right, I can lead off on questions, and it looks like from the chat there's actually quite a few.
Sure, go ahead.
We can lead off with Anna's question, which is a question really, how does something like this handle things like endogenous retroviruses?
Well, it's an interesting question. So the first point of view, the first point to make is in our normal stat tool, as you saw how we make a normal stat database. For normal stat analysis, the way that works is perfect for such a complexity, because a human endogenous retrovirus is going to, a Kmer specific to that retrovirus that matches something in the virus kingdom is going to merge up the tree, and it's not going to stay with the human diagnostic tools. Now, you can argue about whether that's good or bad in reflecting the complexities of biology, but if you know that that's what's happening in analysis, you can isolate that sequence in that manner. Now, in the human scrubber database that Oleg built, I actually believe there may be a few more of those potential retroviral sequences in the database. But when I say a few more, I mean one or two, because remember, again, he purposely did this by subtracting all bacteria and virus and archaebacteria from the human RefSeq Kmer database that he built. So those retrovirus sequences will be gone.
Any other questions? I'm trying to read and type at the same time. Needless to say, I think you will have a number of labs that will be eagerly. I know that the tool has at least been alpha tested by a few labs. I think there's going to be a lot of interest in both running locally as well as having a better understanding of what's running under the hood at SRA. So I'll just leave it open for questions for one more minute.
Hey, Duncan, this is Lee.
Surely.
I know you asked this in chat, but I thought it's worth pointing out, just explicitly asking, how do we get to the singularity image? I'd love to try this out. And then one more follow-up question to you guys is, can this be generalized to things that are not SARS-CoV-2? Because, I mean, there's definitely some obvious applications later on.
Yeah, first of all, let me answer your second question first, and I'm always happy to talk to another cat.
Heck yeah.
So this is not in any way directed at SARS-2 clinical samples, except to the degree that that's, we hope, a very actively being pursued by labs everywhere. That human scrubber database is, again, aggressively removing potential human PII, but it leaves intact any bacterial or viral clinical pathogen sequence read that may exist. So it's not really specific to SARS-2. There is a SARS-2 specific tool based on STAT that's available, but that's for people who want to identify the virus in the sample. It's in some sense the opposite. And I've forgotten your first question, Lee. Can you remind me?
Just the actual applied, like how do we actually get this singularity image? And I mean, I'm saying singularity because it's a lot easier on our shared resources.
Yeah, absolutely. It's I think I can ask someone to add this to the Spheres chat, but this Docker image right here is available publicly to point at. And it's in these slides. I assume the slides will be shared. And also we can send that with the Docker command that I ran, maybe even with a little explanation. Again, that's in the slides, but we'd be happy to share it on the Spheres channel. And I would love people to hit this simply because while I think of this really as a beta version, simply because I didn't package a test with it and put it in a really prominent public location, which would be Docker Hub, it really is the same package. And I'd like people to use it. And if there's any problems, find those problems out now. But I anticipate moving this to a very public location in Docker Hub within a week.
Hey, Ken, this is Rodney. Just to kind of pick up on a couple of things I've been hearing the questions. First thing, do we, Chris, Ken, and others, do we agree that the Spheres Slack channel would be a good place to communicate with folks who want to test out the scrubber and exchange information there?
Depends on that one, but yeah.
Right. So if people are not on that channel that need to be on that channel, let's surface that immediately. And then maybe we can create a little discussion tab there that we can continue to have this discussion. I mean, clearly we're interested in getting as much feedback as quickly as possible. I think it goes without saying that many of you see that this is a very scalable approach. And as we talk about very large numbers of submissions coming into SRA, we would like to have this approach sort of being used. And more importantly, we would like the submitter community feeling comfortable around this approach. So however we can get there as quickly as possible is really key to me. And then second thing, I kind of want to make a shameless plug here. Obviously, Kmer approaches can be used for a variety of different things. They are effectively a sequence search tool. In some ways, you want the sequences to be searched and then scrubbed. And in other ways, you want the sequences to be searched against a high background, right? So we've had teams working on using Kmer approaches since I believe February around SARS-CoV-2. You can imagine them being used for a variety of specific applications, including the detection of very specific alleles within read sets. We're particularly interested in sort of addressing concerns that might arise in surveillance data that comes out of sewage samples and other what are so-called metagenomic sets. And I say all this for my shameless plug that DOE and NIH have started a sort of project together. It's a trans-agency approach to solving sequence search across the entire bolus of SRA data. And one of the sort of first targets will be sequence search that is capable of working against metagenomic sets. So if there are folks here that are kind of interested in that from a development standpoint, I would love to hear from you. This project is in the very early stages. I've talked to some of you a little bit about this in the past. It's a go. And so we'd love to hear that kind of commentary too, because I do think these Kmer approaches can offer us some high sensitivity, high precision methodologies in terms of viral detection, not just for SARS, but for a variety of different viruses. Thank you very much.
All right. Thank you all. Thank you, NCBI. I think, again, I think there's going to be a lot of interest in taking this for a spin, and I'm sure there will be lots of potential test pilots.
Well, great. Thank you. We look forward to you spinning it around.
Yeah. I'm going to say it just started as a big dumb biologist idea And it turns out it works really well, surprisingly well.
But I think, anything that will give labs confidence in, what they're transmitting to repositories would be if they choose to have it done on the SRA side, understanding what's happening behind the curtain.
Yeah.
I think that's very helpful.
I mean, Ken did one simple demo, but we've done the differential analysis of full-on genome alignment versus using our scrubber tool. And the differences come down to some odd, just low complexity sequences, basically. I mean, nothing that you could blast specifically to a human genome. I understand any skepticism, but I encourage you to check it out and provide feedback. It's fast. It works.
Thank you again.